Welcome back to our YouTube channel. Today, we're embarking on a comprehensive journey into the realm of flow cytometry and the immunophenotyping by flow cytometry double negative T cells test. Buckle up as we delve deep into the intricacies of this fascinating diagnostic tool. Pre-test information. But before we dive into the technical details, let's set the stage with some crucial pre-test information. Picture a scenario where a patient walks into a clinic with a complex medical history. Perhaps they've been experiencing recurrent fevers, unexplained weight loss, and persistent fatigue. These symptoms could be indicative of a systemic autoimmune disorder. Now, imagine that this patient has a history of chronic lymphadenopathy, hepatosplenomegaly, or autoimmune cytopenias. These red flags raise suspicion for a condition known as autoimmune lymphoproliferative syndrome, or ALPS. Specification of test. With the clinical context in mind, let's zoom in on the specifics of the immunophenotyping by flow cytometry double negative T cells test. This sophisticated test is designed to identify a distinct population of T cells called double negative T cells, or DNT cells. In healthy individuals, these cells are typically rare. However, in patients with ALPS, there's a notable increase in their abundance. Disease association. Now, let's explore the crucial link between DNT cells and autoimmune lymphoproliferative syndrome. ALPS is a rare genetic disorder characterized by defective lymphocyte apoptosis, leading to abnormal lymphoproliferation and autoimmune manifestations. Specimen used. But how do we collect the necessary sample for this test? Fear not, as the answer lies in a simple blood draw. Yes, that's right. A small vial of blood from the patient is all we need to kickstart the testing process. Components included. Now, let's dissect the components included in the immunophenotyping by flow cytometry double negative T cells test. By leveraging the power of these antibodies, we can distinguish between different T cell subsets and pinpoint the elusive DNT cells. Test report time. But how long do patients have to wait for their test results? The turnaround time for test results can vary depending on the laboratory's workload. However, patients can typically expect to receive their results within a few days to a week after the specimen is collected. Test information. Now, let's unpack some crucial test information. It's important to emphasize that while the presence of DNT cells is strongly associated with ALPS, this test alone is not a definitive diagnostic tool. Abnormal results should be interpreted in conjunction with the patient's clinical history and, if necessary, confirmed through genetic testing for relevant gene mutations associated with ALPS. Conclusion. In summary, the immunophenotyping by flow cytometry double negative T cells test serves as a cornerstone in the diagnosis and management of autoimmune lymphoproliferative syndrome. By understanding its specifications, clinical significance, and limitations, healthcare providers can better navigate the complex landscape of autoimmune disorders. Thank you for joining us on this illuminating journey. Be sure to hit that like button, share this video with your colleagues, and subscribe for more captivating content. Until next time, stay curious and keep exploring the wonders of medical science.